Dear colleagues, we're beginning the session of the Dissertation Council. Before officially starting the procedure, I would like to wish everyone good health, since now the situation is quite hard. We have to adhere to the regulations issued by the Russian healthcare authorities. I'm keeping social distancing right now, and we have to wear masks. I will be putting down the mask while I'm speaking, however. Now, I suggest that we move on with our agenda. Today, I will be referring to the papers that are in front of me quite often, because this procedure and the procedure itself is very important. So I have to comply with the requirements for the defense. Today is the 21st of December 2020. It is 2 p.m. Moscow time. We're beginning the session of the Dissertation Council for the defense of the dissertation by Alan Kaluev. The dissertation is submitted in conformity with requirements for the academic degree of the Doctor of Biological Sciences in Physiology. The topic of the dissertation is biological basis of experimental modeling of CNS processes and human brain disorders using zebrafish. Under the order issued by St. Petersburg State University on the 1st of September 2020, number 7582/1, I, Professor Alexander Markov, Doctor of Biological Sciences of St. Petersburg State University, have been appointed the chairperson of this Dissertation Council. The other members of the Dissertation Council have been appointed by the same order. Let me introduce the members of the Dissertation Board. Here with us, there is Professor Sergei Tsykunov, Doctor of Medical Sciences, Director of the Laboratory of the Physiology of Emotions. Some members of the Dissertation Board have joined us via video conferencing system. They are Professor Yuri Pastukhov, Professor of St. Petersburg State University. Professor Pastukhov, can you hear us? Please don't forget to switch on your microphone. Yes, I can hear you. Everything is okay. The answer is positive. That's good. Professor Yuri Pastukhov, Doctor of Biological Sciences of the Institute of Evolutionary Physiology and Biochemistry. Can you hear us? Yes, I can hear you. Everything is okay. The answer is positive. Professor Maria Tikhonova, Doctor of Biological Sciences, Director of the Laboratory of Physiology and Fundamental Medicine, Senior Research Fellow. She's from the city of Novosibirsk. Professor Tikhonova, can you hear us? Yes, I can hear you. Everything is okay. The answer is positive. You're participating in the session, and I'm honored to present the member of the Dissertation Council from Brazil, Professor Carla Denise Bonon. She's from the Pontifical Catholic University. Professor Bonon, can you hear us?
Yes, I can hear you. Thank you.
Here with us is also Mr. Alan Kaluev, who is the academic degree seeker. I would like to point out some technical issues. I would like to ask the members of the Dissertation Council to switch off your microphones, and also please leave your telephones on a silent mode so that we can contact you in case the internet connection is down. This session of the Dissertation Council is being recorded and broadcast on St. Petersburg State University website, and it is also being interpreted from English into Russian and from Russian into English, which makes my task much easier. Do you have a question? No questions? So I continue with the agenda. You can ask your questions and make comments using the email address that you can find on the official web page. The questions are supposed to be relevant to the contents of the dissertation. The technical personnel will refer the questions to me, and I will read them out during the discussion. As I have already mentioned, the questions are supposed to be relevant to the contents of the dissertation. The authors of the questions are to write down their name, position, and workplace. This is very important. The questions that are not relevant to the dissertation or to the presentation will not be read out. There's something else I would like to emphasize. Under the order on the procedure of granting academic degrees at St. Petersburg State University, hereinafter referred to as the order, this session of the Dissertation Council is duly constituted, provided at least 2/3 of the members of the Dissertation Council are present. The total number is not to be fewer than 4 people. The Dissertation Council consists of 6 people, 6 of which are present, including 4 people who are participating in this session via video conferencing. We can hear and see each other quite well, so we can continue with our agenda since we have a quorum. It's very important. Now, the agenda of the procedure is as follows. The procedure will last for about 2 hours. I will point out the period of time allocated for each item, but these periods of time are by no means obligatory. Number 1, the chairman's presentation about the documents submitted by the academic degree seeker, the presentation given by the academic degree seeker to provide an overview and findings of the research. Next, questions to the academic degree seeker about the presentation. No more than 2 minutes for each questions. I will ask Mr. Kaluev to answer these questions very briefly, and please keep within the time limits. Next, I will be reading out the external reviews that have been obtained by the Dissertation Council, and I would like to ask Mr. Kaluev to provide any answers to the questions if there are any in the external reviews. After that, the members of the Dissertation Council will be taking the floor in turns in order to read out their reviews. After that, I will be reading my review myself. Again, I would like to ask Mr. Kaluev to provide answer to each member of the dissertation board. Next item on the agenda is open discussion. The floor will be given to the members of the audience. There are not many people in the audience today. During this time, I will be answering the questions received online, if any. The next item on the agenda is the deliberations of the members of the Dissertation Council before an open balloting on non-conferring or non-conferring the academic degree. The discussion of the results of the defense is not broadcast. After the deliberations, there will be an open balloting, vote counting and recording the results in the protocol. As far as I understand, I will ask each member of the Dissertation Council how they vote, and each of them will be answering this question. I hope I will be able to calculate all the votes correctly. Next item, making a decision on whether to confer or not to confer the academic degree. After that, we will give the floor to Mr. Kaluev for his closing speech. So, this is the agenda for today. Do you have any comments, questions, or suggestions about our agenda? I have a small question. How do we vote? Do we raise our hands? I will ask each member of the Dissertation Council how do you vote? Do you vote for conferring the academic degree or against conferring the academic degree? And you will answer either yes or no. After that, I will summarize. Is that okay? Yes, I understand everything. Are there any other questions about the agenda? I believe this is a boring part of the procedure, but it is really necessary. The dissertation by Alan Kaluev is submitted in conformity with the requirements for the academic degree of the Doctor of Biological Sciences in Physiology. The topic of the dissertation is biological basis of experimental modeling of CNS processes and human brain disorders using zebrafish. The dissertation was approved for the defense by the order of the Academic Secretary of St. Petersburg State University. The dissertation was completed in St. Petersburg State University. The academic degree seeker has published a number of research papers. He has published 27 research papers. They have been published in peer-reviewed journals, including Scopus and Web of Science journals. There are also 3 certificates of practical use of the research findings issued by Ural State University, Novosibirsk State University, and the National Medical Research Center. Should you have any questions, I will present these documents to you or read them out to you. Moreover, the degree seeker has submitted a full set of documents to the Academic Secretary of St. Petersburg State University. The documents are in compliance with Section 3 of the order, the documents are stored in the dossier. The representative of the Department for Dissertation Council Management has the copies of his documents. Should you have any questions, I can ask them to show these documents to us. So before giving the floor to the academic degree seeker, to Mr. Kaluev, I would like to ask the members of the Dissertation Council if they have any questions to Mr. Kaluev. Do you have any questions to me as the chairperson of this dissertation board? Do you have any questions about the documents submitted by Mr. Kaluev to the Academic Council? We don't have any questions? Good. Since there are no questions, I give the floor to Alan Kaluev. So, the floor is yours. Dear members of the Dissertation Council, dear colleagues, research into normal and pathological brain mechanisms and their treatment is a fundamental task of physiology and modern biomedicine. Mental disorders are widespread. I believe they are even more widespread this year. However, mental disorders are very complex and poorly understood. Poor understanding of CNS pathogenesis is also one of the reasons for the low efficiency of their treatment. One of translational approaches in neurobiology is the expansion of the spectrum of model organisms. This allows us to identify the evolutionary conservative core part of the pathogenic process and the associated biomarkers. The vast majority of works in this area use CNS models in rodents. However, in recent years, a small teleost fish, zebrafish, or Danio rerio, has been used as an effective model in genetics, molecular biology, developmental biology, pharmacology, neuropharmacology, and physiology. The use of zebrafish as a model organism has many advantages, and you can see them on the slide. So, zebrafish can be effectively used for modeling. Therefore, I used zebrafish in order to obtain the objectives of my dissertation. So the objectives of my dissertation are as follows: to study the biological underpinnings of modeling CNS pathogenesis using zebrafish. Next, to identify evolutionary conservative phenotypes, biomarkers, and molecular mechanisms underlining the pathogenesis. This will allow us to translate data from fish to human brain disorders. And finally, my task was to develop and validate new models and biomarkers of CNS pathogenesis in zebrafish. My research also has an element of novelty. In my research, I use a new model organism, zebrafish, for modeling of CNS pathogenesis. I analyze evolutionary conservative mechanisms. I also develop new tests, models, and physiological biomarkers of pathological states in zebrafish. Moreover, my research can be important for future research. I would like to emphasize that the dissertation deals with a wide spectrum of CNS disorders: acute stress, chronic stress, effects of proconvulsant agents in zebrafish, memory stress interplay in zebrafish models, depression-related pathogenesis, and effects of major classes of neuroactive drugs in zebrafish. I am not going to describe in detail each of these aspects. I would like to pinpoint only the major aspects of my research, but I would like to highlight that the spectrum of these disorders is quite wide. I have used a number of methods in my dissertation. We assessed behavioral models. We also used a wide range of neurotropic drugs for pharmacological modulation. I also studied endocrine biomarkers. I also assessed neuroimmunological biomarkers, genome biomarkers, and neuromorphological biomarkers. As an example of using zebrafish to study acute stress, I would like to show you this slide. These are anxiogenic effects of the anxiety pheromone. These data have been published, and many scientists cite these data. Here you can see some very pronounced behaviors, such as freezing, decrease in vertical movement. We also see a lot of total erratic movements. Of course, this response is accompanied by the increase in cortisol. Another example is the withdrawal syndrome. On the slide, you can see a number of drugs that lead to anxiogenic states. Here you can see two lines. I'm sorry, I have to interrupt you. I would like to ask the members of the Dissertation Council that if you leave your room during the presentation, I have to make a technical break. So now we are having technical break number 1. If you have to leave, please let me know. We will have a technical break and then we'll continue. Mr. Kaluev, I'm really sorry. Please continue with your presentation. The first 2 lines show pronounced anxiogenic effects that happen due to withdrawal syndrome. The third line shows the analysis of the cortisol level as a biomarker. Here we see that physiology and behavior go hand in hand, and this is seen in physiological biomarkers. I show in my dissertation that we have an anxiogenic effect here. This is very important for the withdrawal syndrome. In my dissertation, I also studied a wide range of biomarkers that we see in chronic stress. Here we see 5-week chronic daily stress, unpredicted stress. I used a combination of different stresses. You can see all these stresses on this slide. They included bright light, crowding, predator exposure, conspecific exposure, and so on. The fish was exposed to these stresses during 5 weeks. I studied genome biomarkers, endocrine biomarkers, and neuroimmunological biomarkers. What is interesting in this model is that we see affective behavior in fish. Antidepressants change their behavior, of course. When we analyze neuroimmunological biomarkers, we saw the increase of anti-inflammatory and inflammatory cytokines. This is interesting because we see the increase of inflammatory cytokines in humans, but fish demonstrate another aspect here. This is an interesting thing for scientists. We try to understand why there is a difference between fish and humans here. Probably this is due to a high adjustment level to stress in fish. And I believe this difference is worth studying. So zebrafish are not small humans or small rodents. Zebrafish have their own and interesting system in vivo. The same can be applied to neuromorphological biomarkers. We analyzed the data that we received. We analyzed dendritic spines. Stress in zebrafish induced the increase of the number of thin spines. In other words, zebrafish learned how to respond to stress. Stress for zebrafish increased synaptic plasticity. The influence of antidepressants decreased this effect. So it's not an artifact. It's an interesting physiological system. A more detailed analysis, especially in rodents, show the following picture. This synaptic plasticity in stress decreases in many parts of the brain. However, there are some parts of the brains in rodents where this plasticity increases, which is similar to zebrafish. As far as I remember, this happens in hippocampus. So zebrafish models allow us to learn something new, allow us to ask questions. Why is this happening? And not just put a label saying that stress always decreases synaptic plasticity. The picture can become more complex and more interesting. I'm not going to talk in detail about the models of epilepsy, since I'm short of time. I was the first to use nootropic drugs in order to study their influence, their impact on memory. Our laboratory was the first in the world to publish data about piracetam. And you can see the research findings on this slide. Piracetam has an effect on zebrafish. My lab was the first in the world to develop the depression model in zebrafish. We used rezapine, which decreases dopamine. As you see on the slide, rezapine has an effect, has a delayed effect on zebrafish. This effect is pro-depression. It leads to slow locomotion and to the increased level of cortisol. We see this clinical impression in clinical depression in patients. Therefore, this is the state which can be described as depression. This is a depression-like state. My colleague from Singapore proved this model, and they also proved that antidepressants can used as treatment. So this is the model which shows affective pathogenesis. Apart from behavior analysis and other types of analysis, I also studied neurochemical mechanisms of depression. We used fluoxetine and other antidepressants. And in my research, I showed that there are some changes at the neurochemical level. So there are some metabolic changes in the brain. Therefore, behavior, pharmacology, and physiological changes in neurochemical markers can be used to give a broad picture of the pathogenesis. Finally, I would like to give you some data about two drugs that are used for modeling addiction: ethanol and ketamine. Ethanol has a pro-anxiolytic effect in chronic and stress exposure. Here you can see the research findings on the slide. We did not see any changes in the motor functions. Wait a minute, please. Here on the slide, we see that there are no motor dysfunctions under the influence of ethanol. Now a few words about ketamine. Now scientists are studying ketamine because it's an antidepressant. So ketamine is interesting for neuroscience and for neurobiology. And here you see the anxiolytic effect of ketamine. You see here some motor stereotypies, which is interesting in terms of modeling of its effects. What is interesting here is that apart from ketamine, we also tested a number of similar drugs. They all had a similar effect, or they demonstrated specific physiological conservative mechanism. Such effects of ketamine are described in theoretical papers, in clinical papers. We used a number of ketamine-like drugs, and we showed that they can be ranked in terms of their impact. And we see a similar picture in rodents, humans, and fish here. Therefore, we saw a bigger picture. We tested a number of drugs, and our tests demonstrated us new insights into this area. To sum up, my research shows that zebrafish is a highly effective model organism for studying behavioral, cognitive, physiological biomarkers of various mental states related to stress. We identified evolutionary conservative mechanisms of different CNS pathologies. The results that we received can be used for further research, including the development and preclinical screening of drugs to treat mental disorders, especially addiction, depression, anxiety, and other affective disorders. The dissertation has also significantly empowered the potential of zebrafish models to screen a wide range of drugs, including narcotic and hallucinogenic drugs. And to model chronic stress and addictive-related phenomena that are really clinically and socially important. Finally, I would like to thank the members of the dissertation board for reading my dissertation. I would like to thank my colleagues from St. Petersburg State University, from different departments of our university. I would like to thank my colleagues from the University of Experimental Medicine, my colleagues from Almazov Center, my colleagues from the Novosibirsk University of Physiology, and from many other universities. I would like to thank our students and postgraduate students. 95 of them are young people, very skilled and enthusiastic young people. I would like to thank the university for providing support to our laboratory. I would like to thank the technical personnel, and I would like to thank my parents, who are painters, and finally I would like to thank my wife, Natalia, and my daughters, Alexander, Sophia Marie, and Elizabeth, for their support or even not interfering with my work. Thank you. Professor Kaluev, thank you very much. You will have a chance to express your gratitude in your closing remarks, but now I think we should get back to our agenda. I would like to ask the members of the dissertation board if you have any questions. Since there is a pause, I believe that the members of the dissertation board are concentrating. I would like to ask you the following question. I'm interested in the following thing. You said the following thing. During stress, synaptic plasticity increases in zebrafish, in hippocampus. No, it wasn't hippocampus. It was telencephalon. And if we speak about rodents, then the changes happen in hippocampus. As far as I remember, hippocampus consolidates the traces of memory. It's one of the theories. Short-term memory is, to some extent, transformed into long-term memory there. Could you please make a connection between your molecular data and this behavior? Can this behavior be attributed to these two processes? Yes, that's quite right. In one of my papers, I traced the influence of stress on memory in zebrafish. It turned out that stress decreases memory functions in zebrafish. On the other hand, this difference between fish and rodents in terms of synaptic plasticity is also quite important. But BDNF level is also different. BDNF in zebrafish increases in chronic stress, and in rodents and in humans, BDNF level goes down both in brain and in blood. So it's probable that zebrafish is a very ancient species, so probably they have a different mechanism of regulation, different from humans and rodents. The only feasible approach here is to compare two species, rodents and zebrafish. This will allow us to see the differences and the common features, and the point of evolutionary bifurcation will allow us to answer the question, what molecular mechanisms are effective? Thank you very much. Dear colleagues, are there any other questions to Mr. Kaluev? Professor Tikhonova, do you have a question? Dear members of the Dissertation Council, dear Mr. Kaluev, you developed a depression model, reserpine-induced depression model. However, it is believed that a great impact is also done by the genetic component. This is mostly shown in rodents. Therefore, there probably could be a genetic predisposition to depression. This research is mostly done with rodents. Can we use similar approach when we study zebrafish, and is it feasible? Thank you very much for your question. That's a very good question. Yes, there are such attempts. Such studies are done in other labs, namely studies that involve early exposure to stress. But the differences that we see there are of a general feature, are of a general character. Namely, we see high anxiety there. Therefore, we should ask a question. What is applied to zebrafish? Depression as it is, or depression and anxiety? But I believe this is the topic for another dissertation. Unfortunately, there is no motherly behavior in zebrafish. So there are some limitations to our model. There are things which can be applied, and sometimes zebrafish cannot provide some information. Since fish do not show motherly behavior, the model has some limitations, as I have already said. Moreover, zebrafish do not have neocortex, so it's difficult to model processes that involve the functioning of neocortex. One of the ways to validate the model, the depression model, is to use a wide range of antidepressants. We are planning to do so in the future. We are planning to use classical and non-classical antidepressants such as ketamine. I might also study BDNF mechanisms. This is also part of my plans for future research. Unfortunately, there is no specific answer to this question. Does zebrafish have clinical depression or mixed type of depression? Are you satisfied with the answer? Yes, I'm satisfied with the answer. Are there any other questions to Mr. Kaluev? I believe that I would like to ask another question. Professor Kaluev, issues in translational biology are really interesting. So you write about zebrafish, rodents, and a probability of translation these data to humans. Can we use another methodological approach? For example, can we translate data from humans to these models? Thank you very much for your question. This research has been published, unfortunately not by myself, but that's a very interesting idea. The point is, we know about gene candidates for some disorders, and it is very important. There have been studies that show the following clinical impression. The studies involved autism, and they showed candidate gene. This candidate gene was identified. A homologous gene was identified in zebrafish. And after some manipulations, zebrafish expressed autism-like behavior. Well, it's difficult to talk about autism in fish since they don't talk. But in this case, zebrafish demonstrated some dysfunctional social behavior. I believe it proves that the so-called reverse translation from humans to zebrafish is possible, and this is an important area of research. Why is it important? Sometimes it is difficult to make some tests on humans, therefore we can use zebrafish as a proxy screen to test different drugs, different gene manipulations, of so on. So we translate data from humans to zebrafish, make some tests that allow us to assess the effects. If we see some therapeutic effects, then we can make back translation to humans. This idea is extremely important. So zebrafish can be used as a proxy of human disorders, if I may say so. So, are there any other questions? If there are no questions, we will continue with our agenda. When you were showing a slide about ethanol, you spoke about the features of ethanol. You said that it induces stress. No, I did not mean that. I meant that withdrawal from ethanol leads to stress. I would like to ask a question. I believe your research is really interesting. How long do behavioral changes persist in your model? We know there are acute reactions to stress and chronic reactions. What can you tell us about the time period of changes? Thank you very much for your question. The question is really interesting. I'm studying this particular issue right now. We are trying to understand the delayed effects of chronic and acute stress in zebrafish. We are trying to understand how long these states will persist. Right now, we have studied the effects of acute stress. But right now in our lab, we are planning to study the effects of chronic stress. So I will be able to answer your question probably in March, and that's a very interesting question. Are you satisfied with the answer? Thank you very much. I'm sorry I switched off the microphone. I believe we can finish our discussion. Since there are no questions, well, I will make a pause. Okay, there are no other questions. Thank you very much for your presentation. Would you like to take a seat? I'm sorry, I am hyper-aroused. Now, I must read out the external reviews. The Dissertation Council has received 5 external reviews of the dissertation. I will not be, if you don't mind, I will not be reading out the reviews from beginning to end. They contain lots of formal elements, and the degree seeker has spoken about the structure of the dissertation. I will read out the formal part of the reviews, and I will, if you don't mind, I will be talking only about the relevance of the research and probably some questions, if any. Is it okay if I do it this way? Yes, we don't mind. Thank you very much. Wait a minute, please. The first review was written by Professor Mstislavskaya from Novosibirsk Research Center of Physiology and Fundamental Medicine. The dissertation deals with modeling of mental disorders using an alternative model organism, zebrafish. I believe the dissertation is relevant since it allows us to study to model brain pathological disorders. The dissertation describes the impact of neurotropic drugs on zebrafish. The dissertation identifies the main types of impact of these drugs. I would like to highlight that the dissertation makes a great contribution into translational medicine. Moreover, Professor Mstislavskaya also highlights that the dissertation uses a number of methods. This allows Mr. Kaluev to give an insight into this field of science. As I have already said, There is some formal part in the review. I will read out the formal part once only. I believe that the thesis by Alan Kaluev complies with the major requirements set forth in order number 6821/1 as of the 1st of September 2016 on the procedure of granting academic degrees at St. Petersburg State University, and Mr. Alan Kaluev can be awarded the academic degree of the Doctor of Biological Sciences in Physiology. There are no questions in this review. The next review was written by Professor Kaplan from Moscow University. He's the director of the Laboratory of Neurophysiology of Moscow State University. He's a Doctor of Biological Sciences. Professor Kaplan highlights that the topic of the dissertation is relevant. It also has a practical significance. The practical significance of the dissertation is connected with the molecular genetic and physiological similarity between zebrafish and mammals. The author of the dissertation developed pharmacological models of stress-like disorders and also analyzed a number of physiological and genome biomarkers. The laboratory, which is supervised by Alan Kaluev, is the leader in studying zebrafish and its behavior. There are no questions in this review. Therefore, the thesis of Alan Kaluev complies with the major requirements set forth in order on the procedure of granting academic degrees at St. Petersburg State University, and Mr. Alan Kaluev can be awarded the academic degree of the Doctor of Biological Science in Physiology. I would like to highlight again that there are no questions in this review. The next review is from Professor Klimenko, Doctor of Biological Sciences. He works in the Institute of Experimental Medicine in St. Petersburg. Professor Klimenko highlights that the dissertation is relevant since it deals with translational medicine. CNS disorders are widespread in modern world. Therefore, scientists come across and have to solve theoretical and practical issues. And CNS pathogenesis is one of the main tasks today. Modeling of CNS disorders is usually done with the help of rodents. However, the author of the dissertation uses an alternative model organism, namely zebrafish. The tests that have been developed are really efficient for modeling acute stress and chronic stress. Moreover, zebrafish can be efficiently used as a model organism. The model of chronic stress is a new one, and behavioral and different other changes in the behavior can be treated with the help of antidepressants. This proves the relevance of the model that has been developed. The review also contains the analysis of the dissertation, and Professor Klimenko highlights that the research findings make a great contribution into our knowledge about mental disorders. There are no questions in this disorder. So the thesis complies with the major requirements set forth in the order, and the candidate for the degree can be awarded the academic degree of the Doctor of Biological Sciences in Physiology. Finally, there's another review from Dr. Matthew Parker, University of Portsmouth, Great Britain. It's very short, so I will read out it. Brain disorders are highly prevalent human illnesses, yet they're complex and their etiology is largely unclear. Developing novel models of brain disorders and uncovering their mechanisms are an important area of neurobiology and physiology. Dr. Alan Kaluev's doctoral dissertation makes an important contribution to this field. I'm writing this letter to strongly support awarding Dr. Kaluev the degree of Doctor of Biological Sciences in Physiology for his dissertation work presented to the Academic Council of your university. Dr. Kaluev's dissertation comprises an impressive collection of evidence collected and conceptually interpreted him personally that spearheads multiple applications of zebrafish models in neuroscience research. The main goals are listed in the review. The work is topical and acknowledged, as can be seen by a large number of citations to Dr. Kaluev's research papers in literature. The dissertation drawing upon these publications is a comprehensive and well-written volume, which is also logically organized, well-structured, and well-presented. The author's role as a prominent and visible member of neuroscience community is consistent with his demonstrated leadership in the field of zebrafish neurobiology, including his long-term work as the chair of the International Zebrafish Neuroscience Research Consortium. In conclusion, Dr. Kaluev's seminal studies continue to open new important avenues of research in the field of experimental neuroscience and physiology. I believe Professor Alan Kaluev, PhD, as a deserving outstanding scholar to be awarded the higher doctorate degree of Doctor of Biological Sciences in Physiology. There are no questions in this review. So, this is so far for the external reviews. Since there are no questions in the external review, we will continue with our agenda. Now, the members of the Dissertation Council will be talking about their reviews. Can you give me the certificate of practical use of the research findings? There is a certificate of practical use of the research from Almazov Medical University. The research findings are used, or rather the research findings of the dissertation are used in Almazov Medical Center. The research findings are used in lectures and in practical workshops in the Institute of Medical Education. The certificate is duly signed and is duly stamped. Moreover, Ural State University has also issued a certificate of practical use of the research findings of the dissertation by Alan Kaluev. These data are used in lectures and at practical workshops with students majoring in biology in Ural State University. They give a list of subjects which make use of the research findings. They include applied biomedical research in toxicology, pharmacology, and pharmaceuticals, alternative models in preclinical studies, laboratory animals in preclinical and biomedical studies. All in all, the research findings are used in 7 courses which are provided by this university. The certificate is duly signed and duly stamped. There is also another certificate of practical use of the research findings issued by Novosibirsk State University. The certificate is duly signed by Professor Tulupov. He certifies that the research findings of the dissertation by Alan Kaluev are used in educational processes in this university. The research findings are used in a number of subjects taught at this university. Therefore, I have presented the certificates of practical use of the research findings. And now I think we can continue with our agenda. First of all, I would like to give the floor to Professor Carla Denis Bonan. We'll be happy to read your review of the dissertation.
Thank you very much. So, good afternoon. I'd like to thank the St. Petersburg State University and Professor Alan Kaluev for the invitation to participate in the Habilitation Dissertation Committee. It's a great honor and a pleasure for me to be part of this committee. Professor Kaluev's dissertation summarizes his research for the last 2 decades. It deals with multiple aspects of zebrafish behavior and pathologies, including modeling affective disorders and studying common drug effects on zebrafish. When Professor Kaluev started his work on zebrafish, they remained largely understudied, and in my opinion, his pushing and innovative use of these species helped the field to grow in the last decades. In the dissertation, he summarized several papers that, for the first time, introduced the effects of common and widely spread psychoactive substances on zebrafish, thus characterizing the fish typical behavior in different states and contributing for successful translation between human, rodents, and zebrafish. Furthermore, Professor Kaluev was one of the first scientists introducing complex models of neurobehavioral disorders in the field of zebrafish studies and deepening our understanding of the models, studying behavioral and molecular alterations, as well as developing new methodologies to model human disorders. The present dissertation work is organized as group chapters, each with a focus on a specific theme in modeling psychiatric conditions or drug testing. The first chapters focus on summary of overall methods used in the work. The last chapters focus on testing wide range of psychoactive substances in zebrafish and allows deep description of common neurobehavioral spectrum, including serotoninergic, GABAergic, and glutamatergic systems and associated behavioral operations. Finally, chapters 3 to 6 study several models, both physical and pharmacological, including depression-related models, proconvulsant drugs, and validates tests on zebrafish special methods. In summary, Professor Kaluev's work is a complete, comprehensive, and innovative study on zebrafish. Based on my reading of his dissertation, I think he is also an outstanding scientist, both enthusiastic and skilled and extremely productive. I strongly believe that as Alan's work continues, he will see many other insights in the neurobiology of zebrafish and successful translation of this work to human neurobiology with development of new therapy models, drug targets, and understanding of evolutionally conservative pathological aspects of the disorders. So it's my opinion that Dr. Alan Kaluev fully and undoubtedly deserves awarding him the degree of Doctor of Biological Sciences by St. Petersburg State University. So thank you very much for your attention.
Большое спасибо вам за...
Professor Bonan, thank you very much for your positive assessment. Since there are no questions in the review, I would like to thank Professor Carla Denis Bonan. I will ask the next member of the dissertation board to read out their review. I'm following this document that I have in front of me. I would like to give the floor to Professor Pastukhov. Please, we are ready to listen to you. Thank you very much. I would like to say that the academic degree seeker has presented his dissertation to the Academic Council. He mostly deals with zebrafish. However, in his dissertation, he also focuses on the differences between zebrafish, rodents, and humans. Therefore, the analysis is really comprehensive. The author of the dissertation has written a really comprehensive research. It is way better than many other works in this field. The dissertation also focuses on CNS disorders. It also focuses on biomarkers that can be used for preclinical studies. Moreover, Mr. Kaluev has developed a number of neuropharmacological models of stress-like behavior. He also analyzes the effects of nootropic agents on behavior and on memory. I can talk at length about the research findings, but I'm going to be brief. We have been participating for 15 years in an international forum which deals with the stress and behavior. The forum was organized by Professor Kaluev in St. Petersburg. Sometimes the number of international participants was as large as 700 people. So we are familiar with Alan Kaluev's works. Right now, our laboratory deals with the molecular mechanisms of mental disorders. Before that, we also studied the role of mediators in different models of mental disorders. Therefore, I would like to say that I have a number of questions. I have read your dissertation, and the following questions have arisen. What about temperature fluctuations? Do they have an effect on stress level? I will continue reading my question. I'm interested both in chronic stress and in acute stress. So, did temperature fluctuations have any effects on the behavior of zebrafish? Did you see any biochemical or genetic changes in the brain under the temperature fluctuations? So I have a couple of questions about the dissertation. I have analyzed the text of the dissertation very thoroughly, and I believe that the author of the dissertation has achieved all the objectives of the dissertation. He was able to develop a model in zebrafish. He also identified the evolutionary conservative core of different behavioral disorders. He has also identified some specific features of stress-like behavior in zebrafish, and these data clearly correlate with clinical data. And finally, zebrafish can be effectively used in preclinical studies, which is proven by the dissertation itself. So, to sum up, the dissertation of Kaluev, Biological Basis of Experimental Modeling of CNS Processes and Human Brain Disorders using Zebrafish, complies with the major requirements set forth in the relevant order of St. Petersburg State University. And Mr. Kaluev can be awarded the academic degree of the Doctor of Academic Sciences in Physiology, Specialization number 030301. The dissertation is compliant with Section 11 of the above-mentioned order. Thank you. Thank you very much. I give the floor to Mr. Kaluev to answer your question. Professor Pastukhov, thank you for your question. I had some time to think it over, so I would like to share my opinions about it. I believe that environmental factors are really important for any model organism, including zebrafish. But unlike many other model organisms which can be used for research, zebrafish is a unique model organism. It is one of the few species whose natural habitat is about 10 degrees expanded. In other words, zebrafish can live in cold rivers of India and in warm swamps in Bangladesh. So zebrafish has a very wide natural habitat. The optimal temperature or the range of optimal temperature is 12 degrees, which is quite rare. So this allows us to use zebrafish as a model organism which is resistant to temperature fluctuations. Nevertheless, zebrafish has its own preferences. Given a chance, zebrafish will go to a warmer place. They know their favorite temperature, and this is their preference factor, and we can use warm water as a reward, for example. But to answer your question, I would like to give you this information. In my research, I control temperature like many other factors. We use the same temperature for the environment, for the exposition tube, and for the novel tank. The temperature is 25 degrees Celsius in the tank. So I take into account this factor and I minimize temperature variations that might have effect on zebrafish behavior. But zebrafish biology is unique. It is more resistant to temperature fluctuations. Professor Tikhonov, are you satisfied with the answer? Yes, I'm satisfied with the answer. Thank you very much. I would like to say that this was Professor Pastukhov. I must say, I was planning to follow the alphabetic order, and unfortunately the first was supposed to be Professor Alexei Yegorov. So, Professor Yegorov, the floor is yours. Thank you very much. My review is posted on the official website, so I'm not going to read it from beginning to end. The doctoral dissertation should open up a new avenue into research, and I believe that this is exactly what this dissertation does. He solves a fundamental task and he opens up new avenues of research. He has developed an alternative model. Moreover, he has obtained unique data, namely responses to different types of stress. He developed different new models of stress-like behavior. What is shown in the dissertation is unique data that makes a great contribution. I believe the dissertation opens up possibilities for future research in this area. I believe that the idea presented in the dissertation is unique, and I believe Professor Kaluev has been working in this area for many years, for about 15 years. And I see that you have been developing this area of research for many years, and this dissertation is the summarization of your long-term research. I also like the methods that you used. I believe that the dissertation is really well structured. You mostly focus on zebrafish. However, you have done other types of work as well, as far as I know, in your lab. You have published a number of papers in high-ranking international journals. I believe it's high time you became a doctor of biological sciences. This dissertation defense is just a formal part. Still, I have some questions and comments, but they are debatable. Question number 1. On pages 7, 8 and some other pages you write about CNS pathogenesis, which is not quite accurate. I think we should rather talk about pathogenesis of CNS disorders or diseases. On page 41 you write about the withdrawal syndrome, and I believe that the fish was exposed to caffeine and diazepam for a short period of time. Is it enough for the withdrawal syndrome to develop, or can we attribute the changes in their behavior to different post-intoxication changes? My third question, did you take into account gender differences, which are usually taken into account in rodents? To summarize, the dissertation opens new avenues of research The dissertation by Kaluev is an important contribution to science and research. I believe that the dissertation, Biological Basis of Experimental Modeling of CNS Processes and Human Brain Disorders using Zebrafish, complies with the major requirements set forth in the relevant order of St. Petersburg State University, and Mr. Kaluev can be awarded the academic degree of the Doctor of Biological Sciences in Physiology. Thank you. Thank you very much for your review. Thank you very much for your comments. I really appreciate them. I know that you are involved in translational research yourself. You carry out preclinical trials using animals. So I would like to answer questions very thoroughly. I agree with your comments relating to the use of terms. I believe this is connected with the translation of terms. The English term CNS pathogenesis sounds okay. However, when we translate this term into Russian, we should add such words as disorder and disease in the Russian language. You also asked about the withdrawal syndrome. Thank you for this interesting question. In modeling, in developing the model, I took into account the half-life period of the drugs that I used. So there was no accumulation of the drug, And however, the syndrome withdrawal was evident. We had to do some scaling here, and we did it quite successfully. And here physiological biomarkers were very important. For example, cortisol level is an important physiological biomarker here. The cortisol level increased when the exposure to the drugs was stopped. The increase of stress hormones reveal that it was an anxiety-like state. There were also some other biomarkers, such as C4s. They are activated in stress and in withdrawal. Finally, there are some papers which model withdrawal syndromes. Several papers have been published dealing with this issue, and they prove that here we deal with affective disorder, not with not with the effect of the toxicity. I believe further research will help us to differentiate between these two states. But the overall model is quite accurate. There's one exception here, caffeine. Caffeine did not induce withdrawal syndrome in fish. Probably caffeine has its own specifics. It stimulates humans at first, and it can lead to panic attacks if taken in large doses. However, it is anxiogenic. It has an anxiogenic effect when it is given to zebrafish. So zebrafish don't like coffee, but fish like nicotine, for example. Now your question about gender differences. That's a very important issue. It is raised regularly in different types of research. Our latest research is not part of the dissertation. However, my latest research shows that there are some gender differences in terms of response to stress and in terms of response to pharmacological treatment. This is a very important issue which must be studied thoroughly, and my colleagues and I are taking first steps. I believe scientists in Brazil also conduct such research. This is a very relevant issue, and we are just beginning to study gender differences. Professor Yegorov, are you satisfied with the answers? Yes, I'm absolutely satisfied with the answers. Now I give the floor to Professor Tikhonova. Dear Chairman, dear members of the Dissertation Board, I am not going to read out the review from beginning to end, since it is posted on the website. However, I would like to highlight some points. The dissertation is really topical. Modeling of mental disorders and translation of these data are cutting-edge research right now. And I believe that Mr. Kaluev has raised a number of important issues, and he was one of the first to start using zebrafish as a model organism. We need a technical break number 2. A technical specialist has reminded me that you shouldn't leave. Oh, I just pressed the wrong button. I would like to remind you about the procedure. Oh, please continue, Professor Tikhanova. It's okay. Mr. Kaluev was one of the pioneers of this research, namely the modeling of mental disorders in zebrafish. And right now, Mr. Kaluev is a leading scientist in this field. His works are cited by many scientists. There are more than 300 citations to his papers in Web of Science and Scopus. This is a highly valuable research. I believe that the research, his dissertation, is complete research. It opens up new avenues of future research. Mr. Kaluev has been organizing international conferences for many years, and these conferences provide a platform for scientific debate. At first, he organized small conferences. I was happy to attend one of the first conferences organized by Professor Kaluev. Now, his conference is a big international forum which is hosted by many countries such as Japan and Taiwan. Some time ago, I visited a conference in Japan, and I would like to highlight the high level of presentations made at this conference. I have some comments and remarks about the dissertation. However, my comments do not change the overall positive assessment of the dissertation. You have presented a lot of data in your dissertation. However, the analysis of this data is not really thorough, is not really elaborate. I wish you would have given a more detailed interpretation of the data. And here I have a question. Do your models comply with the general models of mental disorders? Namely, the models, or rather the criteria for models developed for rodents. Can these criteria be applied to zebrafish, or do we need to develop new criteria? There are also some smaller remarks and technical remarks, but in general, My remarks and my critical remarks do not change the overall positive assessment of the dissertation. So the dissertation by Mr. Alan Kaluev complies with the major requirements set forth in order on the procedure of granting academic degrees at St. Petersburg State University, and Mr. Alan Kaluev can be awarded the academic degree of the Doctor of Biological Sciences in Physiology. The dissertation is compliant with Section 11 of the above-mentioned order. Professor Tikhonova, thank you very much. Please answer briefly. Thank you, Professor Tikhonova. I would like to say that I believe that validity criteria are really important. In my dissertation, there are some biomarkers such as motor retardation and the level of cortisol allow us to come to the conclusion that fish have similar states, states similar to those in humans and rodents. This proves the etiological validity of the model. Moreover, the use of rezapine and different other antidepressants in our research allow us to talk about the predictive validity of our model. Model of rezapine-induced stress was used by my colleagues in other universities, and they used my model successfully. So it also proves that my model has a predictive validity as well. But still, fish and rodents are two different species standing apart, and the data translation is important here, but it's not that easy to do. I don't think that we need to develop specific validity criteria for zebrafish, since here we see more overlaps, more evolutionary homologs than differences. But probably the aquatic specificity should be taken into account when developing the model. Unfortunately, not everything is clear here. Zebrafish has not been used in biology for such a long time, but a way of 1,000 miles starts with one step, as we know. The more we think about it, the more clarity we will have on this issue, and I would like to thank you for your question. Professor Tikhonova, are you satisfied with the answer? Yes, I'm satisfied with the answer. Now I give the floor to Professor Sergei Tsykunov. Dear members of the Dissertation Council, dear Chairman, this research is really topical, since research of mental disorders is a really complex issue. Mental disorders are widespread. It is really important to understand the mechanism of these problems, and modeling is extremely important here. It is extremely difficult to model human mental disorders using animals, but it is important, otherwise we would not be able to understand the mechanisms of mental disorders. Another issue here is treatment of mental disorders. Treatment that exists today is sometimes not efficient. because treatment is not always efficient and effective. This dissertation allows us to develop new therapeutic treatment and also to develop new therapeutic targets. The dissertation, in my opinion, gives a copious description of the material. Moreover, it validates the models that are developed. Mr. Kaluev writes at length about the correlation between behavioral changes in fish and in humans. It is difficult to talk about direct correlations, but the correlations Mr. Kaluev was talking about look quite valid and credible, because freezing is always freezing. Another thing I would like to highlight here, there are some neurochemical changes in the models. Sometimes we can say that we proved something, or sometimes we say that we have come up with something new. But this research identifies new approaches to analyzing the data that we have. I was really interested to read the data about neuroplasticity. Neuroplasticity is regarded sometimes as adjustment. Probably neuroplasticity is an attempt to adjust to the environment. But if neuroplasticity persists and has the feature of neurodegeneration, then neuroplasticity can be regarded as a pathology. So neuroplasticity has a dubious character, and this is well shown in your dissertation. Moreover, you used common antidepressants, which is also important for your research. It is really important to provide validation of the model. What is important here is to study the use of new drugs and the repurposing of older drugs, and the models developed in the dissertation allow us to do so. The dissertation applies new methods and provides a copious description of the material. I believe the dissertation complies with the major requirements set forth in the relevant order, and Mr. Alan Kaluev can be awarded the academic degree of the Doctor of Biological Sciences in Physiology. Thank you. Thank you very much. As far as I understand, there are no questions in your review. Now, I'm supposed to read out my review. I have an advantage here because a lot has been already said, so I believe I can be very brief. I will try to talk slowly so that I can be interpreted accurately. This defense is a big celebration for me. I am the head of the Department of Physiology of St. Petersburg State University. Ivan Pavlov graduated from this department. We all remember this scientist. we always remember the work done by Ivan Pavlov. Moreover, Professor Alexei Ukhtamsky, who developed the research of dominant behavior, also worked at our department. I graduated from the Department of Higher Neurological Activity, and I remember tests in fish that were conducted when I was still a student. We studied reflexes and behavior in fish, but unfortunately this topic was not developed further. But now we see the reappearance of this research thanks to your dissertation and thanks to your studies. What I like about your research, where I see the relevance of your research, you use different models that can identify the physiological mechanism and the pathological aspects. One of these methods is the evolutionary approach. So you use models that stand at different stages of evolution. Another important method is the study of pathologies. It's the pathology that allows us to see the essence of the dysfunction and the essence of the disorder. Moreover, you have studied a number of pharmacological drugs. You wrote about chronic stress, acute stress. You conducted some important tests, such as a new tank, such as a novel test tank. All in all, your dissertation gives very important research findings. I'm writing about your dissertation, about the structure of your dissertation in my review. I really liked your dissertation. I believe it is a well-structured and high-quality research. I asked my question during the discussion, so I'm not going to repeat it. So finally, the dissertation by Alan Kaluev complies with the major requirements set forth in order issued by St. Petersburg State University on the procedure of granting academic degrees in St. Petersburg State University, and Mr. Alan Kaluev can be awarded the academic degree of the Doctor of Biological Sciences in Physiology. Mr. Alan Kaluev has answered all the questions. I believe we should proceed with our agenda. Now I must ask the members of the technical staff the following question. Thank you very much for your work. Are there any questions that you received via email? There are no questions in the email. Due to the lockdown measures, there are few people in the classroom today, so there are no questions from the audience. Therefore, I suggest that we continue with our agenda. I must ask the following question. There are some members of the dissertation board who are participating in the session via video conferencing. So, does anybody have questions about the video conferencing system? I will make a pause to give you a chance to ask your questions, should you have any. So I make a pause. Thank you. There are no questions about the video conferencing system. The next item on the agenda is open balloting and voting. The voting may consist of two parts. Should you need a technical break to discuss the results of the procedure, we will have one. Okay, I repeat. We can switch off the broadcast and make a technical pause in order to discuss the results of the dissertation among the members of the dissertation board. If you do not think that we need a technical pause, then we can move on to voting straight ahead. I don't think we need a technical break here to discuss the results. Thank you. I think we can start the voting process. Okay. I believe we can begin the open voting. Okay. We're beginning the open voting on the issue of conferring the academic degree of the Doctor of Biological Sciences, Specialization 030301 Physiology. Let me remind you that the decision of the Dissertation Board on conferring the academic degree is positive, provided more than 50% of the council members voted in favor of conferring the academic degree to the degree seeker. So I will be asking your opinion. Okay. Professor Carla Denise Bonan, how do you vote? Yes or no?
Yes.
Thank you very much. Professor Yegorov, how do you vote? Yes. Professor Pastukhov, how do you vote? Yes, I vote in favor of conferring the academic degree. Professor Tikhonova, how do you vote? Yes. Professor Tsikhanov, yes, I also vote in favor. So, dear members of the Dissertation Council, out of 6 members of the Dissertation Council, 6 members of the Dissertation Council have voted in favor of conferring the academic degree. Nobody abstained from voting and nobody voted against conferring the academic degree. So, Mr. Alan Kaluev is conferred the academic degree of the Doctor of Biological Sciences in Physiology, Specialization 030301. Dear colleagues, there are some members of the Dissertation Council who are participating via video conferencing system. Do you have any questions? Or comments about the procedure of the defense? No, we don't have any comments. Thank you very much. That's a very important issue in the procedure. Now, I'm glad to give the floor to Professor Kaluev for your closing remark. You can say whatever you want here. Thank you very much. Thank you very much, dear colleagues. Thank you for your support and for your trust. I would like to emphasize the following thing. When I was asked who I should invite as the member of the dissertation board, I had my specific idea. I wanted my colleagues to be in this dissertation board. I wanted my colleagues whose opinion I respect. Unfortunately, the university did not include all the scientists that were on the list in the short list, but I really respect all the colleagues who are here. I would like to thank you for your comments and for your interesting questions. And I would like to thank you for discussing this research with me, because after this procedure I will have to think about a lot of things. I would like to say a few words about translational approach. I would like to highlight two aspects here. When we use zebrafish, we can talk about two types of translation. I'm talking about direct translation now. I call it the so-called inside translation and outside translation. The inside translational approach allows us to assess the evolutionary bridges between different species. It allows us to develop new types of treatment, and it gives us new insights into the underlying mechanisms of different disorders. This is the inside translational approach, and zebrafish allows us to use this approach. On the other hand, zebrafish allows us to use the so-called outside translational approach. Rodents, apes and dogs cannot be used to study 10,000 drugs per day, but models with zebrafish are highly effective and highly productive. I don't want to say that we must test 10,000 drugs daily in each lab, but I'm talking about potential capabilities. Sometimes it is necessary to test a lot of drugs, or sometimes it is necessary to repurpose the existing drugs. Sometimes it is necessary to identify new effects of drugs. In this case, zebrafish is really effective. So this translational aspect, this translational potential of the model is also important. Finally, I would like to use the opportunity and say the following things. I would like to congratulate all the members of the dissertation board. I would like to congratulate my colleagues. I would like to congratulate young scientists on New Year and Christmas. I hope we will have new ideas in the new years, and I hope that we will continue our studies. I hope we'll be going forward and forward at a great speed. Thank you very much for your assistance and for the interesting ideas that we shared here. Thank you. Professor Kaluev, thank you very much. We will celebrate New Year and Christmas, and on behalf of the members of the Dissertation Council, on behalf of your students, I would like to congratulate you. This year is not a good one, but for you, this year brought success and possibility for future research. I believe that we should congratulate Professor Kaluev on the successful defense of his dissertation. Thank you. According to our tradition, we must give him a round of applause. So let's give Professor Kaluev a round of applause. I'm looking at the technical personnel here. I believe we can officially put an end to our procedure. So this is the end of the session of the Dissertation Council. I would like to wish everyone to be healthy and to stay well. And I hope you will have new hopes and new plans in the new year. Thank you.